Welcome to your lesson on lipids and nucleic acids. Notice that lipids are also made of carbon, hydrogen, and oxygen. The major difference here is that because you're going to have these huge, big, long chains like you see in this picture here, you have way more carbon, so you have a high carbon content, and you also will have a lot less oxygen compared to hydrogens. You can see already just in these little pictures here, this little thing has 3 carbons, it has 3 oxygens, but it has more than 2-to-1 hydrogens. So there's no 2-to-1 ratio like there is in carbs. Exact same types of atoms, you've still got carbons, hydrogens, and oxygens, but you're not going to have that 2-to-1 ratio. What's the point of a lipid? Where you have sugar being a quick source of energy, you store it as a glycogen if you're animal or starch if you were a plant. You have all those glucose kind of just tucked in together and you can just chop them off. This is a longer version of energy storage. In other words, longer time. You can store way more energy in these molecules because really the energy is stored between these carbon bonds. So if you have way more carbons, you can store a lot more energy, but it's also going to take some energy to break these up so that you can turn them back into sugars. But we'll talk about that in a completely different unit as we move forward in the course. But just so you know, it is a long-term energy storage solution. It's also very valuable in other ways as well. It's not just about energy. It's also about insulating your body. It's about cushioning your internal organs. You need to have fat in there. And it's about regulating cell activity. A lot of your hormones are based on fats. So they're fat-based, kind of like a steroid hormone that you'll see shortly. Okay. So you want to make sure that you've got fat in your diet. There's good type of fat and there's bad type of fat. You need good fat in your diet. Not only that, but it also gives you a feeling of fullness. It's what satisfies you in a meal. You don't want to cut out fats completely. You might want to reduce harmful fats and eat good fats, but you still need fat in your diet. So let's go through and we'll talk specifically about its structure. Fats, no surprise, don't mix with water. And the reason for that is they are nonpolar. Because they end up being these huge, big, long molecules, it's not a lot of oxygen stealing electrons part-time from a carbon. There's not a lot of polar covalent bonds. And so because of that, you tend to have an equal distribution. Overall, that molecule is neutral. It's not charged in any way. It's not polar. And so because of that, it will not interact with water. If like dissolves like, this is nonpolar and water's polar, you're not going to dissolve it. And that's where you're going to have the layering of oil due to its density above water. It'll sit on top of the water. So nonpolar, therefore hydrophobic because it's insoluble in that water. Right? It's formed the exact same way that we formed our sugars. We form it through a condensation reaction. What's condensation reaction? That's the dehydration synthesis. That's when we removed the water in the last picture so that we could join those molecules together. Okay, I'm going to do it exactly the same way here. Just so for the sake of understanding short forms, you can write this molecule out like this with 1,000,000 carbons, or you can do the short form. What you do is you take all these middle CH2s and you just say, well, how many are there? In this particular picture, there happens to be 7 of them. So I can acknowledge this group on the end here, which is our carboxyl, and then I can acknowledge the fact that there's 7 of these little repeated units here. And then I just acknowledge what's on the very end. It happens to have a CH3 group on the end. Okay, so that's the short form way that we can do things. So when later on when you see it down below, and you see that it's a 16 here, it just means it's twice the length of this molecule. It's 16 instead of 7. When you are joining these together, what you have to understand is that you're not just joining together one fat to another fat, because fats actually don't form chains. Instead, what happens is you make one complete fat. And you do that by joining this molecule here, called a glycerol. You're going to see a lot of GLYC molecules, glycogen, glycerol, all sorts of different things. You even have the linkage that we just made in the sugars. If I scroll back, this linkage that we just made here is called a glycosidic linkage. Like, there's lots of glyc words. It's up to you to try to keep those different terms straight. So in this case here, you have a glycerol. The OL at the end of the word tells you it's a version of alcohol, and alcohols tend to have these OH groups. So just in general, just so you know, that's why it's called a glycerol. It's the way that the functional groups are arranged. I could take those functional groups and move them into a slightly different orientation, and I would have to call that molecule something else. But in organic chemistry, that's a glycerol, alcohol version of glycerol. It's got 3 carbons, 3 hydroxyls, and the rest are hydrogens. what I'm going to do is I'm going to take a single, what's called a fatty acid. It's not a fat, it's a fatty acid because of this carboxyl group makes it acidic. And I'm going to do the same thing I did before. I'm going to chop off an O and an H on the one end and just an H on the other. So I'm going to have an oxygen joining directly to this carbon here. I'm doing a dehydration synthesis just like I did before. And what it ends up looking like is this here. So we're going to remove two H's and an O to make a water, and that's going to combine this carbon here directly to that oxygen here, and you can see it in this picture. There's that carbon there directly to the oxygen of the glycerol. But I have 3 spots I can do that, so I can do it 3 times. So here I've got 3 fatty acids joined to 1 glycerol to make this complete lipid or this complete fat. There's lots of different versions of fat. This is the triglyceride version. Triglyceride. Tri because there's 3 fatty acids attached to the glycerol. Okay. I don't have to have a fatty acid at everything. I could put a phosphate group there, and we'll see that with phospholipids shortly. That's how your cell membranes are made. I could put something completely different at one of those things. I don't have to have all exactly the same fatty acid. I don't have to have them 16 long. I don't even have to have them where they all have where they all have a single bond. I don't have to have them all single bonded. I can have them double bonded. And as soon as you do a double bond, you introduce a kink to the molecule. And I'll show you that as well. Okay. But this in general is called a triglyceride again because you have 1 glycerol, and in all 3 hydroxyl spots, you've decided to put in a fatty acid. Pay attention to the types of functional groups that are being joined together. When we did sugar, it was 2 hydroxyls joined together. Now that we're doing fats, you have a hydroxyl on the glycerol, but this here, that's not a hydroxyl. It's a COOH. This is a carboxyl group on the end here. So I've got a hydroxyl here and a carboxyl interacting, and those are the ones that are going to form my bond. The name of that bond is also going to be important. It's called an ester linkage. Memorize those. Which groups join together and what was the name of that bond. If you can lock that away in your memory for all 4 types of macromolecules, you're going to do well on this next little quiz. So it's a hydroxyl group and a carboxyl group. We're dehydrating it, so it's dehydration synthesis. And in this case, I've done dehydration synthesis 3 times. So when I go to do my molecule, I have 3 waters produced to make 1 fat. So I've got some pieces of paper here, and they're going to simulate fats. Okay? Fats do not form polymers. You will never get 1 fat physically connected to the other, but what you will get is them sitting on top of each other. Okay? If it is a saturated fat, because you've got all of those carbons and they're completely full up with hydrogens, we would say that they are saturated with hydrogens. Okay? Saturated means full of hydrogens. So then unsaturated then means that not every carbon has its full complement. If that's the case, if I have a carbon and it's not filled up with hydrogens, what'll end up happening is you'll get a double bond between the carbons because they still have to be satisfied. They're just satisfied by each other rather than being satisfied by hydrogens. And you'll draw that in a second. But here's how that ends up playing out in terms of fats. These pages are all meant to represent separate fats. In this case, saturated fats because they're all straight molecules. Those carbons will all be saturated and they'll just form these nice straight long molecules. Okay? If that's the case, they're going to be nice and tightly packed together. And what you should know from particle theory is that the closer I can get those particles, the more attraction I'll have between each individual portion. So this forms actually a really, really nice because it's between, intermolecular forces. It keeps them together. And in fact, it's so good at keeping them together because they're so flat that I'm going to form a nice tight solid. But as soon as I'm missing some hydrogens and I start forming those kinks in the molecule where the carbon actually, the chains begin to bend and twist, what I end up with then is 2 unsaturated fats that when they connect, they're not forming nearly as much of a tight bond. There's not as many parts touching. And so because of that, they're more likely to flow over each other. Way better than I can even simulate it with the actual paper. So here we have the solids, okay? So as the solids they're not going to slide off quite as well. But if I have, even on this scale, the larger scale, but if I have these bent molecules, because they have less points of contact, have to hold that one better. They'll easily fall off each other. How does that play out? It means that your saturated fats form solids and your unsaturated fats form fluids. They form liquids. So here we have then, again, stacked. And because they can all be flat, because they're straight linear molecules, I'm going to be able to get solids at room temperature. And so you'll see your saturated fats are in your butters, your lards, your stiff butters. I mean, they still fall apart pretty easily into liquids when you heat them up a little bit, but they're at room temperature, they'll be the solids. These are the guys that clog your arteries for that exact reason. It's because they're nice straight molecules. So these are not the good fats, these are the bad fats. When we go to unsaturated fats, having that little kink in the molecule, now obviously here we short formed it so we didn't draw all 3 fatty acids, but imagine that we've got a similar chain poking off of this part of the glycerol as well. Okay. We didn't even bother to draw the glycerol properly, we used a box to show that. But if you have missing hydrogens, you still have to have 4 bonds for this carbon, but now you're going to allow the bonds to be satisfied by each other rather than filled in by hydrogens. And then again, because of that, you'll see a kink in the molecule. And here's an over-dramatized version of kinks in molecules. You get all these kinks in the molecules. They will no longer lay flat because you get all the kinks. And then when that happens, you have the liquid version. Unsaturated fats are going to be your liquid oils. These are the ones that are good for you. Just best guess based on what the word mono versus poly means. When you look at this diagram here, do you think it's a mono unsaturated fat or is this a poly unsaturated fat? In your diet, it says it's a mono unsaturated fat. It's only one kink. Whereas if it's a poly unsaturated fat, there's many of these kinks. So again, it's the unsaturated fats that are going to be good for you. We have this weird fat, though, that we use as a spread for toast and whatnot, and that's margarine. It's not quite as solid as butter, but it's certainly not a liquid oil, and it is made from liquid oil. And what they do is they take the liquid oils that have these kinks in them, and they bubble hydrogen gas through so that it straightens them out slightly. Not completely, but enough to make it a more solid version of oil. And that's why it's called hydrogenated vegetable oil. So not quite as solid as a butter, but enough to make this vegetable oil spreadable rather than just pouring it onto your bread. You probably have also heard of trans fats. Well, trans fats were really, really great when they first discovered them. And the idea of a trans versus what we would call cis fat, C-I-S, cis versus trans, is just the direction that the bend happens, whether it's up versus down for those bends. Let's say you are in the business of making French fries and you've got a big vat of oil. Anyone ever had to do that job? When you drop those fries into the vat of oil, you can only use that oil for so long before it becomes rancid. And you can't serve rancid oil to your customers. Okay. So over time, after you've used it so many times, you have to take all of that oil and you have to dump it and replace it. Well, the great thing about a trans fat is that it lasts longer. So it was really, really great when you're starting to fry all this stuff up because you could get a longer shelf life and a longer use life out of your oil, which was wonderful. But then they found that these trans oils, the ones that they had influenced, because they had that weird kink going in the wrong direction, it was trans versus the normal cis, that it was just as bad in your arteries as a normal saturated fat was. It was just as sticky and caused just as bad of clogs. So now you'll see on the chip bags and all sorts of stuff like that over the last 10, 15 years, once they realized trans fats wasn't as great as they thought, they started putting things on there and saying no trans fats because people were worried that they were clogging arteries. Make sense? All right, so what are the other types of fat? We're almost done here. Wax is a type of fat. So your ear wax or the wax that lines the cuticles of your fingernails. That's made from fats. It's just a different version of it. It's big, long carbon chains. Then you get phospholipids, and this is an important one for our course because we're going to look at the structure of the cell membrane. I should mention that the waxes, they are solid at room temperature. Room temperature is about 21 degrees Celsius, but it doesn't take very much to liquefy them. So 45 degrees is a very low melting point. When we talk about the structure of plants later on, we'll be talking about waxes again, and we talk about leaves and how they're constructed. And you'll see that the wax that's on the leaf is going to form a nice waterproof barrier because of the polar versus nonpolar nature of it. All fats are nonpolar, so all fats are hydrophobic, meaning they do not interact with water. So if you want to have a nice protective layer, just sort of like what's on your skin, where you have a nice protective layer of oil being produced by the sebaceous glands on your skin, you're going to have a nice waterproof layer. It is important for you to know the structure of phospholipids, again, because we are going to talk about cell membranes, and you want to know how cell membranes are put together. It's basically a glycerol with 2 fatty acids, but in that third position, instead of having a fatty acid dropping down there, instead you've got a phosphate group there. If you were to look back on the first page with the functional groups in it, and you looked for a phosphate group, you'd see that it is a phosphorus atom surrounded by a bunch of oxygens. Phosphorus only has a few locations where you can actually add in oxygens. So you end up with all these bonds, but you end up with a whole lot of unsatisfied oxygens. Oxygens that are very electronegative, very electron hungry, and ready to rip electrons from anything because, again, this is the godfather, okay, in terms of electronegativity, okay? So because of that, as soon as I add in a phosphate, I get this negative end of the molecule. So even though these are nonpolar, this is a very polar end of the molecule. That means that I've now got a phospholipid, phospho because phosphate, lipid because it's a lipid, that you have one part of it that loves water and the other part of it that wants nothing to do with water. So I end up with a hydrophilic head, hydrophilic loving water head, and a nonpolar or hydrophobic tail. Why do I care? Because this explains why I can take a pile of phospholipids, throw it into water, and I can immediately, without even asking it to, I can immediately make this lipid bilayer that ends up happening. Bilayer meaning 2 layers. And so you'll have the heads towards the water, both outside and inside the cell, and you'll have the little tails that'll turn towards each other for comfort. They kind of hide together. If you just have the normal ones where we just had the glycerol with 3 fatty acids, they'll all be pulling up their tails to try and stay away from the water. That's why they'll sit on top of the water. But in this case here, I can be in the water, but I'm going to be hydrophilic out, hydrophobic tucked in, okay? We're going to form 2 layers though. So you have hydrophilic head sticking towards the water here, tails inside, and then hydrophilic sticking out here, hydrophobic sticking inside. And you got to think of it, even though this is drawn as 2D, you have to think of it as a 3D sphere now. So I've got this layer with water inside, layer of fat, water outside. It's amazing because we don't need to encourage it to do that. It behaves like this because of its structure. If you get the structure right on the molecule level, you automatically get the macro level structure right. You're going to see that a lot with proteins as well. Put the amino acids in the right order, and you'll get a perfect protein every time. It'll fold and naturally do things on its own. So that is your phospholipid bilayer because you've got a polar head and a nonpolar tail. Very last thing, looking at steroid hormones here. You've heard of steroids before. You've heard of hormones before. Probably didn't know that the vast majority of your hormones are steroids. Testosterone is a steroid hormone. That's generally the one that they're trying to use to get them to build up muscle mass. Estrogen, progesterone, all these guys they're all made of their modified fats like this. The difference though is I don't have these big long chains. Instead, I have these carbon rings. So in general, the reason there's numbers here is because this is a 6-sided figure, 6-sided, 6-sided, 5-sided. And then you have this little functional group sticking off the bottom here. Now, if you want a different steroid, you'll have the same sort of carbon rings, but you might have different functional groups sticking off, and that's what allows it to behave differently. So estrogen and testosterone may be very chemically similar, but they're just different enough that they interact with your cells differently. We usually call testosterone the male hormone, and we call estrogen the female hormone. The fact is that everyone has testosterone and estrogen, but whether you're male or female depends on which one is the dominant hormone. So as you go into puberty and your body begins to do all those secondary sex characteristics and your body is changing and developing, males produce a lot of testosterone, which is going to influence their muscle size, their actual height, all that kind of stuff, the secondary sex characteristics like growing a beard, all that kind of thing. And then estrogen affects women because we have a lot more estrogen compared to testosterone. It'll affect us in the opposite direction. So it'll lead to fat development within the body, certain fat placement. And that's why you tend to have this is the male physical characteristics versus female. It's how much of each of these hormones did you have while you were developing. There are some people that are going to be testosterone deficient, young men, maybe because The gonads where these are produced are not going to be producing enough of the testosterone, so they might have to be supplied more testosterone in order to build their bulk. Some people who are cheating in the Olympics may also take testosterone as a steroid to help them to produce more muscle mass and all that kind of stuff. Extra testosterone tends to lead to aggression, and so it's not necessarily a good idea to be taking more than what you need. You need enough to develop, but not enough to make you a scary Hulk, basically. So that's the basic idea. Steroid hormones, we'll come back to them when we talk about the actual homeostasis unit. So I just want to quickly run through how to draw the fats, because again, you're being tested on this tomorrow. The test will be drawing glucose plus glucose equals a maltose. I demonstrated that in the last lesson. Then you'll have a question where you'll have to do the fat. So you'll show glycerol before a fatty acid, and then you'll show glycerol joined to all 3 fatty acids to make a triglyceride. So I'm going to show you that now. And then you'll also have a protein to do. But I'm going to pick the easiest amino acid to put the proteins together, the easiest sugar, which is glucose, to put the sugars together, and then a simple fatty acid here. So when you're going through and doing this activity, again, it's pretty simple, step by step. Just run through and get it done. What elements are on glycerol? Carbon, hydrogen, oxygen. Not difficult. So it's meant to be just easy, step by step. But you need to be able to draw these guys. So the one that I want you to memorize is this one right here. Butyric acid. So when you go to make your fat, you're going to use a glycerol, which is the same for all of them. That's the one that was above. And butyric acid. The but part, the but part, just tells me that there's a couple of, you know how we said there was those CH2s in the middle? So you've got your carboxyl. And you've got this methyl group here. And then you've got in the middle 2 times CH2, right? That's the butyric part, is that there's 2 of those guys in the middle. If you had 1 or you had 3 or more, or whatever, every time you add in more of those CH2s in the saturated fat, you change the name so that it acknowledges how many of them are there. Okay? So butyric just means that there's these 4 carbons in a row that are there, right? Always make sure you have the carboxyl, have the 2 in the middle, and then have that CH3 at the end, okay? But make sure you can draw this in all different orientations. That's what it's going to ask you to do down here in question 2. So don't just draw it once, draw it and then rotate it, because you're going to find that the easiest way to do this picture is to actually have this guy completely flipped so that the carboxyl is on this end. So you need to know how to move and orient that guy around. That's why I'm asking you to do that. And of course, I want you to memorize it. So I want you to be able to see it in all different orientations. When we go to put it together, this is the exact question that you'll have on the quiz. You're going to make a triglyceride using butyric acid. So drawing a triglyceride using 3 butyric acids. Now keep in mind, you have to have a glycerol to join them to. So here's how we draw it out. We're going to do our glycerol first, but because we got these fatty acid chains coming off, we're going to want to make sure that our glycerol is really long. So there's lots of space between the carbons so we can fit things nicely. And you're probably going to want to draw this quite a few times to get it stuck into your brain. So the glycerol, if you go back up, is this guy here. 3 carbons, 3 hydroxyls, everything else is a hydrogen. Actually, you know what we'll do before you do that? Let's do before and after. I'm going to show you the formation of it so that you've got the idea of what it looks like before. So in this case, we can do a small glycerol. So 1, 2, 3, OH, OH, OH. Everything else is a hydrogen. Sometimes I find it's easier if you draw the bonds and then you just fill in with the letters. Okay, so that's my glycerol. My butyric acid then, I'm gonna want to have the carboxyl group is what's going to interact with the hydroxyls here. So I'm going to flip the molecule from what you saw on the page before. I'm going to draw myself the carboxyl. So double bond to the O. How many of the CH2 groups do I need for a butyric? 2, 1, 2. And then you have that final CH3 group, right? So 1, 2, 3. Everything is just hydrogens. One mistake that people tend to make is they get hydrogen happy, kind of go crazy with all these hydrogens everywhere. Don't be that person. Make sure that you're still considering the bonds, because lots of people try to put an H here, and that's just not correct. You can't put an H there because there's not enough space on the carbon to add another one. You've already got 4 bonds, right? So make sure that you're not hydrogen happy when you're doing these things. So a reminder again, we're doing a dehydration synthesis reaction, so we know that we're going to be ripping off the value of H2O. Now, because you're showing the formation of it, you would actually technically have to do 3 butyric acids, but I can let you get away with just saying times 3, because if you've drawn it correctly the one time, I don't need you to draw it correctly all 3 times. So this would be before, and then for the formation, you're going to show me what it looks like afterwards. What's it going to look like? It's this plus this times 3, and it's going to go to this. So here's where we want to have our little bit spread out glycerol. So 1, 2, 3. Okay. Now I know I'm going to have the O's sticking out, but I'm not going to have the hydrogens attached anymore because I will have already removed them. Then I know that I'm going to be joining directly to this carbon right here. So I just start drawing this part of the carbon chain, 4 carbons in a row. So I'm going to go 1, 2, 3, 4. And that's the same for all 3 fatty acids that are being joined. 1, 2, 3, 4. And now I just need to, next thing I would do is do my double bonded O so I don't lose it. So it's double bonded O, double bonded O, double bonded O. Everything else is a hydrogen. And then I'm done. So I like to do my lines first. It's faster. 1, 2, 1, 2, 3, 1, 2, 1, 2, 3. That's where all my hydrogens are going to go, and then I just fill them in. So it's not too tough. It just takes a little bit of practice to try to remember what the silly little details are. So if that's my first attempt, I'm a little annoyed with the fact that my hydrogens seem to be a little too squished in there. So on my second attempt, I would just make my glycerol a little wider so that if these are stretched a little more, I have more space to play with. And then you just get it perfect. Okay. So again, the question will be the formation of these things. So you have to be able to draw it before and afterwards. What's missing? I haven't completely formed this sucker yet. Plus 3 waters, thank you. Okay, we had to remove the 3 waters in order to get this to work, so you have to acknowledge the fact that that is also produced. There's other questions, though, that you're going to have associated on the written part of the test, and so you're going to want to remember that you've got hydroxyls interacting with a carboxyl. I don't need you to label it on there when you're doing the drawing test, but I need you to know that information. So the name of this bond is an ester linkage. If you want to call it ester bond, I'm okay with that too. That's not a problem. But you need to know it's an ester. So you got to be putting all that kind of information together along with the drawings, and the drawings just take practice and paying attention to details. And that, my friends, is the end of the lipid lesson. We'll continue with nucleic acids at another time.